Darrell, DNA in plants and grapes in particular, you don't sort of think of plants and DNA.
I guess most people don't. Those of us who are paid to think about it quite a lot. But most people think about testing for quality. There's other chemicals that people test all the time. So a Brix test for soluble solids is often when you're trying to test how much sugar is in a grape to see if it's ripe, or people test for phenolics to get an idea of the flavor of the juice. So DNA is just another chemical that's in every cell, but it gives us other information. So rather than telling us whether a grape is ripe, it tells us hereditary information, so what variety of grape it is, and we can link that to information about where it will grow best, or what kind of wine it'll make in the end. So yeah, it's another chemical that gives us information.
So you're taking a whole lot of the guesswork out.
It is, I guess. Yeah. I mean, or don't guess, we test, and then we take away the assumptions that are often made, and it actually can give a definitive result about the genetic background.
So I guess if you're buying vines to plant in your vineyard, you don't have to wait for 3 years to make sure you bought the right ones.
Absolutely, yeah. And that's quite a big problem, is buying a vine and then saying that, well, this has a leaf shape that looks like Sauvignon Blanc, so we assume it is, and then you propagate up 10 hectares of vines and on the assumption that that's what they are. But when we look at molecular markers, in the DNA, we can actually say this has a piece of hereditary information that is unique to Sauvignon Blanc, so we can be confident. And we can test that just based on a sort of tiny little punch from a leaf.
Are you looking at this to get new varieties?
Yes, we are. So in an associated technology, what we do is we started by looking at the fact that New Zealand actually has a high incidence of new varieties forming. And what I mean by that is new clones and new types of a variety are usually identified by finding a branch in the vineyard that looks like it's doing unusually well, or has unusually big grapes or something like that, and then they take a cutting from that and they grow an entire vine off of it. And so, for example, Pinot Blanc is a white variety that was established from a red variety, Pinot Noir, which stopped producing that red pigment. And so that kind of change is easy to identify in the field, but if you're looking for something like disease resistance, it's very hard to identify without a devastating disease coming through your crop and ruining them all. So what we do is we've studied what causes vines to change like that. Why would a new branch suddenly be different from the rest of the vine? And that's to do with how the genetics, the DNA, slowly changes over time. And we then take samples of the vine, and in tissue culture we separate out individual cells, and then we monitor those cells as they undergo these small genetic changes, and then using growth media, we grow them up into individual plants. So we get one plant from every single cell, and that means that we have a unique new plant that the entire plant has those small changes. So if a change, like in the example of the red to white, if that occurs in one of the cells, we end up with a whole new plant that would have white grapes. So it's the same thing that happens naturally in a vineyard, but because we're doing it a cell at a time, we can monitor the precise genetic changes that occur, and we can generate an entire population where each vine has a small, subtle change in it.
Be a good time saver.
Absolutely, yeah, because we can actually monitor what has happened, what has changed in the plant as it occurs. We can say as soon as the plant is maybe a sort of 5 centimeter young plantlet, we can extract some DNA, see if anything's changed, and then even look at what genes have changed. We can say this is a change that occurred in a gene that is likely to affect the yield of grapes ultimately. And so we can pick the ones that look like they'll be winners and grow those up into full vines, rather than hoping you find something and generating thousands of vines with a possibility that some of them will have something unique in them.
Darrell, the end user, how long before what you're doing here in the lab actually ends up in people's vineyards?
So we've created a pilot population of about 200 vines already, and those we're speaking with the New Zealand Wine Growers Association, who've been quite excited to access them as potential new varieties. We are now just starting to expand that population, so we'll be producing several thousand new vines that each have subtle changes. So ultimately, it doesn't take that long to produce the new vines. It only probably takes a year or 18 months. but the phenotyping, which is looking at what has changed in the plant and actually assessing whether it'll be good for New Zealand's environment and whether it'll be an optimal crop and whether it'll grow, whether it'll produce good wine actually ultimately, takes a bit of time. So that we'll do in combination with nurseries and wine growers to actually assess this vine looks good in the lab, it appears to have a better yield, but is the wine it actually makes any good to drink?
So I guess this is no different to people working on getting a new wheat variety or ryegrass variety?
Yeah, it's no different to finding this thing naturally. So we're not genetically modifying the plants in that we're not introducing any foreign DNA, we're not manipulating their DNA physically, we're just watching what happens in the natural, how cells naturally change, but rather than hoping we find them in a field of wheat, to take your example, we're actually screening them as it occurs in real time.
How did you get into all this?
Well, my background was in molecular biology, and I studied at Stellenbosch University in South Africa, so I was in the middle of the winelands there, but I was actually looking at forensic DNA markers. So the markers that we look at to discriminate one variety from another are essentially the same markers that are used in paternity testing or any other forensic testing in humans. And we were looking at markers that can discriminate one population of abalone, which we call in New Zealand power, from another, because there's a lot of smuggling. In fact, sort of 80% of the abalone in South Africa are ultimately smuggled out to the east, and the police want to be able to seize a shipment and be able to say this when the people holding the power say no, this is from China originally, be able to do a DNA test and say no, these are from a population from Cape Town, and we have a DNA test to show them. So it's much like paternity testing.
So DNA is definitely very, very much a part of science.
Absolutely, and my passion is really bringing it to agricultural science. It's been used in human and health sciences in a long time, so there's lots of DNA tests available for hereditary diseases. If anybody who knows their blood type ultimately has had an indirect DNA test done on them because they know some genetic hereditary information about themselves. And so it's really been very powerful for people to be able to know, for example, some of these devastating diseases that are late onset, whether you're a carrier for them, and so that you could prepare or take lifestyle choices which would actually affect that. And now we're trying to introduce these technologies to the agricultural industry so that people can actually get genetic information about the plants that they're growing and actually use these to produce better crops.